Alright, hello everybody. My name is David Poplin, and I am going to be giving my presentation on quorum sensing signaling response system in gram-negative bacteria. Thank you for participating. So I wanted to start my talk about the difference between gram-positive and gram-negative bacteria. The way that they classified these two different bacteriums was by gram staining. This gram staining allowed them to classify bacteria species based on their chemical and physical properties. And so, as you can recall, gram positive have the thick peptidoglycan cell wall. This is going to be the more violet color. Gram negative, on the other hand, has the thin peptidoglycan cell wall that obtains the pink color during the gram staining. Some gram-positive bacteria are cyanobacteria, salmonella, and E. coli. On this topic, we are going to be focusing on gram-negative bacteria, specifically V. fisheri and V. harveyi. Now here is just a representation of gram-negative and gram-positive bacteria. You can see here that the peptidoglycan cell wall is very thin for gram-negative bacteria and thick here for gram-positive. This is the main point that I want to bring up about the different gram-negative and gram-positive bacteria, because it's going to play a role in some of the ways that they quorum sense. The good old Bonnie Bassler. So Bonnie Bassler is called the bacteria whisperer, and she has claimed this because of her extensive research on quorum sensing. Bonnie Bassler has a Bachelor of Science in Biochemistry that she received from the University of California, Davis. Then she received her PhD in Biochemistry from Johns Hopkins University. After that, she has been at Princeton University since 1994. Now, I'm sure Schneller has a lot to say about Bonnie Bassler, so we will save more about her for his input. Now, what is quorum sensing, and why do we care about quorum sensing? So quorum sensing is a way that bacteria can perform cell-to-cell communication. It is a system of stimuli and response correlated to population density. So bacteria will use quorum sensing in order to figure out the population density of their bacterial colony. So when quorum sensing occurs, they release these autoinducer molecules, which are extracellular signal molecules. These molecules will collectively modify gene expression and behavior of the colony in a whole. Now, some say that quorum sensing is an important evolutionary milestone that ultimately gave rise to multicellular life forms. So I wanted to talk about some important reasons why bacteria might want to quorum sense. Now, the first thing I wanted to bring up was symbiosis, which is any type of close or long-term biological interaction between two different organisms. The second thing would be virulence, which is a pathogen's or microbe's ability to infect a host organism. Motility is important because this is the ability of a bacteria to move independently using metabolic energy. Anaerobic production is also very important because this can help with the treatment and prevention of bacterial infections. Biofilm formation is also an important factor in quorum sensing, and it is done by, it occurs when cells become embedded with a slimy extracellular matrix composed of extracellular polymeric substances. Quorum sensing uses signal molecules called autoinducers. These autoinducers are responsible for the production, release, and the ability of surrounding bacteria to detect these molecules. Now, as the population of the bacteria increases, they are able to synthesize more autoinducers, so the concentration of autoinducers increases respectively. These autoinducers are able to monitor and track the bacteria's population. They can also alter patterns of gene expression and regulate physiological activities. It is important that they are able to diffuse freely throughout the bacterial membrane. That way they are able to be produced within the bacteria, diffused into the extracellular component, and be able to be received by a receptor molecule in another bacteria. These autoinducers are synthesized from S-adenosylmethionine, or aka SAM. I'll definitely go more into detail about SAM and how these autoinducers are synthesized, but for now, this is a brief introduction. So the most common class of autoinducers in gram-negative bacteria are going to be the acyl homoserine lactones, or AHLs. Here's a small chemical structure of an N-acyl homoserine lactone in a gram-negative bacterium. So the way that these autoinducers are produced is through a protein called LuxI, which is the initiator protein, and they synthesize these AHLs. Now, in order for these AHLs to be received through other bacteria, there's another protein called LuxR. The LuxR protein is a transcriptional factor that activates expression of the Lux operon in the presence of AHLs. LuxI type proteins have a specific LuxR type protein that binds its AHL. So here's a couple examples of gram-negative autoinducers in a couple different bacterias. Now, as you can see, that to the left, they have the same or similar homoserine lactones. So these, here are a couple examples of some gram-negative autoinducers. As you can see that on the left over here, they have these different bacteria all have the same or similar homoserine lactones with varying R groups to the right. Now, these gram-negative autoinducers are species specific to each autoinducer signal molecule, meaning that only the bacteria that make the signal can respond to it. This is called intraspecies quorum sensing. This is important because if we wanted to use a V. harveyi autoinducer to, let's say, for example, bioluminesce a V. fisheri bacteria, it would not work because the autoinducer is species specific, and the receptor will not be able to receive the signal molecule. So in tonight's talk, I'm going to be focusing on two different types of bacteria that use quorum sensing. The first bacteria that I'm going to be talking about is Vibrio fischeri. This bacteria is a gram-negative marine bacterium. It is a bacterium that is reliant on another organism. So it is a symbiotic bacterium, and sometimes the cells can only survive on decaying organic matter. Now, as I stated earlier, the most common class of autoinducers is our acylhomoserine, or AHL, which is produced with our Lux-I type synthase. The main AHL used in Vibrio fischeri is our N3-oxohexanoyl homoserine lactone, which is easily diffusible through the cell wall. This AHL is used as a proxy for cell density to activate luminescence. Bioluminescence is controlled by a small set of genes known as the Lux operon. So the amide linkage formation from 3-oxohexanoyl and methionine groups and the lactonization of the methionine side chain are the two reaction steps involved in the formation of homoserine lactone autoinducers. So here we have our 3-oxohexanoyl ACP, which is the asocarrier protein, plus our S-adenosylmethionine, which is our SAM that I talked about earlier. These two molecules will react to produce our N3-oxohexanoyl homoserine lactone that is going to be our main AHL in Vibrio fischeri. This reaction is catalyzed using the autoinducer synthetase. So here's an example of how our bacteria will use this Lux system to create and receive our autoinducer. So at high cell density, autoinducer concentration increases in the cell's local environment, increasing the number of binding sites of AHL to the cytoplasmic receptor Lux-R. So up here, as you can see, the Lux-I is the initiator that creates these these signal molecules, the autoinducers, which are diffused through the cell membrane to reach the extracellular components. So here is at low concentration, and here's an example at high concentration. At high concentration, you can see that other bacteria here are able to receive the autoinducers. Here's an example of how the autoinducer will bind to the Lux-R, creating a Lux-R AHL complex that acts as a transcriptional activator of luciferase gene LUX-CDABE. So once the Lux-R and the autoinducer are binded, they bind to an area upstream of the operon called the Lux box. After this, the expression of luciferase results in bioluminescence. This allows for bacteria to count their numbers and turn on genes that are useful when bacteria are acting as a group. So typically, these, at low concentrations, the bacteria is not going to be able to express luminescence. But once they reach high concentrations, then each bacteria is better in numbers and are able to create bioluminescence. So here is a slide that will give the visual of Vibrio fischeri communication. So here we see at low cell density there is no bioluminescence, so no light occurs. This is because there are low concentrations of these autoinducers. Now, as high cell density occurs and the cells start to accumulate, they are also producing a higher concentration of these autoinducers, as you can see. So, as the population increases, so does the production of these autoinducers, which allows the bacteria as a whole to express the genes luciferase, which will allow bioluminescence. The easiest example to talk about for Vibrio fischeri is the symbiotic relationship between the Hawaiian bobtail squid. Now in this, the development and growth of both organisms occur as they cooperate with each other. So Vibrio fischeri cells utilize the pili to interact with the squid's host, and then they start to mature on light organs of the Hawaiian bobtail squid. So what they do is they erase the shadow that would normally be seen as the moon's rays strike the squid, which protects the squids from its predators. The squid in turn provides the bacteria with shelter and a stable source of nutrients, which is usually sugars and amino acids. So here is a picture of the Hawaiian bobtail squid. They are only about that big, so they're very small organisms. What's very interesting is they have these light organs that attract the Vibrio fischeri, and the Vibrio fischeri will then go into these light organs and start producing these autoinducers. Once the accumulation of these autoinducers reach a point of favorability for luciferase gene to be expressed, they emit a wavelength that counteracts the wavelength of the light organs, which eliminates the shadow of these squid. Now this allows the squid to go out and hunt at night and not be able to be seen from the starlight or light from the moon. Now, once these bacteria emit this wavelength, they are technically not going to emit any more wavelength, or they are depleted. So the squid have these pumps that will pump off the old or used Vibrio or V. fischeri, and will then be able to obtain new bacteria. Here's another photo of our squid They're actually really cool. I wish I had some in my fish tank back here. That would be really nice. But it's not the way it goes, I guess. The second example I'm going to be talking about involves Vibrio harvii. So Vibrio harvii is a free-living marine bacterium that utilizes two different autoinducers. Autoinducer 1 is a species-specific homoserine lactone. So this autoinducer is very similar to the one used in our Vibrio fischeri example. Now, what's unique about Vibrio harvii is it utilizes another autoinducer called autoinducer 2. Now, this autoinducer could be an interspecies communication molecule. Now, this language, per se, could have arose before the split between gram-negative and gram-positive bacteria. So here's a great representation of Corrin sensing in V. harvii. As I stated earlier, that autoinducer 1 is a homoserine lactone, and autoinducer 2 is not. So each autoinducer was detected by its own sensory system. And as you can see, A1 is detected by a protein called LuxN. A2, on the other hand, is detected by two proteins called LuxP and LuxQ, which work together to detect and send the information from the autoinducer 2. Now, LuxQ and LuxN are called bacterial two-component signaling proteins, and they are both very highly conserved. Now, they have transmembrane domains that connect the inside of the cell to the outside of the environment. As you can see here, here's LuxN's transmembrane domain, and here is LuxP and LuxQ's. Information is sent through a phosphorylation cascade that involves histidine and aspartate residues. So, at low cell density, LuxN and LuxQ are kinases that phosphorylate and transfer the phosphate group to protein LuxU, which then will transfer that phosphate group to LuxO, which will then in turn turn off the luciferase gene, which will not create any light. Now, at high cell density, the autoinducers bind to sensors, and the sensors change from being kinases to phosphatases. The phosphate will then, in turn, flow back into the system and dephosphorylate LuxO, releasing it and allowing luciferase to be expressed. Hence, bioluminesce. So here is the LuxS reaction using our S-ribosyl homocysteine. So here we have our LuxS reaction that initially starts with our S-ribosyl ribosyl homocysteine. And as you can see, that our Lux S is used as an enzyme to cleave this molecule to yield our 4,5-dihydroxy-2,3-pentane dione and a homocysteine. Now the autoinducer 2 formation starts with this 4,5-dihydroxy-2,3-pentane dione and yields a 2,4-dihydroxy-2-methyldihydrofuran threone, and through a hydrolysis reaction will then yield 2-methyl-2,3,3,4 tetrahydroxyl tetrahydrofuran, and through the elimination of 2 waters and the addition of a borate, we then get our 2-methyl-2,3,3,4 tetrahydroxy tetrahydrofuran borate, which is found to be our autoinducer 2 molecule. Now, as an overview, Lux S protein synthesizes our pentane dione that is used as a precursor to obtain our AI2. Now, the AI2 controls many genes in many different bacteria. So this autoinducer can be used for the intercommunication between different bacteria. And that is all I have for you guys. I thought this was a cool slide to end it on. We've done something very similar to something like this with our GFP lab. And I wish nothing but the best for all of you guys and hope you have a great finals week. Bye. Now this is Stella. Eventually, we might be able to put the luciferase gene into Stella and she can shine like a star. Thank you. Bye.